I gave George one. And that's cool.
Yeah, it was a cool one.
Yeah. Did you do your window shade thing?
I pulled it down last night. I mean, I guess I pulled it down more. I ended up just pulling the lights to be super bright. So, well, this is what... I'll stop the streaming.
Oh, the lights need to be super bright to get your...
They're on the quarter system there, so...
So that's too bad about the work. Yeah. Maybe one thing I want to do is, if they have their reviews there, right? Yeah, and then maybe share them with people.
Sure.
Yeah, right.
Alright, when are you going?
Next Wednesday.
Oh, good. That's very soon. Did you find
My lease.
enough room to live in over there?
Yeah, like hell. We got this like in-law faculty house.
Okay.
So,
A little bit more than a box.
yeah, no, it's pretty big.
Thank you.
Thanks.
So is your live stream all set up?
Yeah, I couldn't get the display to work properly. Like because there's two screens, it sets up for just that screen. Like you can size the things up. And so when I put this up, it like fills the whole display with this, even though I told it only to take part of it. So the camera overlaps with some of the stuff. There we go.
So there's no like output when you go to, like in that program I use Camtasia.
Yeah.
Have a menu for what video you want to capture.
Yeah.
The one that's leaving on a cable or the one that's local.
It's asking me to capture like a window. There's some other way to capture like the little screen, but
You're not gonna have that.
no. Yeah, I started by
Now
Summertime, yeah.
summertime 10 a.m. Monday.
How are you going to post this in minutes?
Yeah.
Alright, so I mean 50, up to 65 years.
Oh, bagel. What?
I mean, I don't want to be a pig, but I also don't want to do it so that 2 minutes after I start on that. Yeah, I mean, what's going to happen is
I would like to talk to
think, in fact, my car is.
Oh, you have it, shit.
Yeah.
You're gonna flow.
I briefly followed
Well, it's more fun if you have somebody.
my best friend from California to fly out.
But, you know, you got a lot.
So I was sort of hacking I realized that
Just said that.
I think of them as my wall stickers. So
Our phones have
So,
next one's
is this one of those things where somebody drives it out? Or is this a
I guess the area is like outside what core is. I'm out of that business sense. Evil. So talk doesn't offer anything.
free labor.
Well, some circumstances.
That's all right, but at least you have skills.
So
This doesn't work out.
neuroscience, but I feel it's too arbitrary. It's
funny,
many
is it?
sides. And cognitive this is circles and also neural estimate of science. It gets so out of hand.
Right. Yeah.
I feel like every area has to fall.
This is the I thought it was one. Do we have like the
It seems
faculty is four.
Next time this way.
yes.
Yeah.
Yeah, sure. Sorry. Okay. Hi. So good morning. Thank you for coming to Zarrar's defense. We're very excited and happy for him, and we hope it goes well. He'll give a talk, after which we'll have time for just a few questions, and then we're gonna, and then we go into a private session, which can take anywhere between 15 to 30 minutes. Yeah, and then Greg will, his advisor will introduce Zarrar before he starts. So thank you.
And you're all invited up to my lab, which is a 310 SSS building behind us afterwards for a toast if things go well, or the toast if things don't go well. So anyway, I'm just going to say a very few words about Zarrar because I I really want this time to be his time to present his research. But you know, he's got quite a notable, I looked this morning to see how he's doing in Google Scholar. He has 44 papers, 5,600 citations with an H index of 23. So he's outperforming many junior faculty at other universities. Zarrar came here a while ago to work with Jeremy Gray, a colleague of ours who's now at Michigan State, and I was very happy when Zarrar decided to come into my lab after he left. I think he's, you know, I say this with nothing but sincerity, the most generative student I've ever seen. He has more ideas before breakfast than I have in a year, which sometimes can be a bad thing. But I think in this case, it's a very good thing. He's going to present today 3 experiments, which is really just a small amount of the work that he's done, particularly a small amount of the work that he's done in these various studies. They have real, I think, important methodological innovations, which he's not going to emphasize today, but I want to emphasize because he really worked these things out, and watching him do this was quite amazing. Clever ways to remove shared information, clever ways to measure directed connectivity, and the application of neural nets to fMRI data, which I thought was extremely clever, particularly, and this is work he's not going to talk about, how he compared the output of the neural nets to behavioral studies that he did both in living subjects and also in MTurk, whatever subjects those happen to be. So, you know, it's been a real honor for me to have Zarrar in the lab. He's always full of ideas. He's always pointing out the newest papers for me to read. It's exciting to be around him, and I've learned as much as I've taught him. When he's finished, presuming this goes well today, he will be hanging around for a couple of months as we get the rest of these papers out, but then he's moving on to Columbia, where he's going to do a postdoc with Daphne Shahami, who's going to be here, I guess, sometime this coming fall to give a talk. So with no further words from me, I'd like to turn it over. Zarrar is going to talk on his defense, localized computations, and distributed representation. Zarrar?
Thanks very much. So today I'll be presenting some of the work I've done. Hopefully it's not too early. Greg kept that saying, like, you know, it might be too early for people, but so thank you for coming. So I wanted to start by talking about how we began thinking about this in terms of the views that people had in the field about brain organization, and in particular between 2 competing views that are seen as very opposing. And so I'll be illustrating those views here on this schematic brain. The circles here indicate different brain regions. And so the first view that I'll talk about is a localized organization, where each region is associated with a particular function. So here I've shown 4 visual categories on the left. Each of those categories, like faces and letters, is associated with a particular brain area. And sometimes it's also known or used to associate with a modular organization of the brain in psychology. In contrast, a distributed organization posits that it's the combinatorial or the combination of activity across a whole set of areas that encodes a function. So here I'm showing how faces is associated with a set of brain activity in the brain. Each category would have a different set of brain activity, like letters and so on. And now these differences are really historic. So they come from the beginnings of neuroscience with like Gall, Florenz, etc. And also there's somewhat personal, as I began some of the projects here, and some of the work that I began, not shown here, I looked at information in terms of entropy. And when I began to look at information in terms of entropy, I saw information was everywhere. So information was, for faces, was present in every part of the brain. Every part of the brain was relevant for processing faces. Now that was a striking contrast to some of the work that was done in our lab before that found that there were very specific areas of the brain that were relevant. So, you know, we wonder then in the lab, like, well, why do we have such huge differences between some of the measures that I began with and some of the measures that we've historically been doing? And so one thing that we came upon was that maybe these differences in the way we view how the brain is organized is driven by methodological differences in the approaches we take. So for instance, for the localized organization, that could be driven by a psychological model we might have, where we have some sort of black box models where each box is associated with a particular function. And it's very natural for then people to say, okay, this box that does X is associated with very particular areas of the brain. And so the methods, brain lesion and brain activation studies that are used to support it, take a very localized or narrow view of where stuff is in the brain. In contrast, if you're interested in a distributed organization, you might begin with a computer science maybe background or model, where you have an idea that the brain is this parallel distributed organization. And then you might use pattern analyses, these multivariate analyses, that look at the brain in a very distributed way. And so we can see then that two different ways of thinking lead to two different ways of maybe understanding how the brain actually is. And so maybe these different aspects of understanding the brain reflect actually the same phenomenon. And so we'll be exploring that here in this talk, looking at a third unifying perspective where we consider the brain as a connected network, and where each region can accept any information. So what happens in this model is that we think that each individual area of the brain does these local computations, which means that each area does its own thing, its own operations. And then that local work that's done is shared via connections in the brain and that information then spreads throughout the brain, leading to a distributed representation. So what we end up observing, which started as something localized, is something that looks very distributed. And so we can see we can sort of connect these localized views and these distributed views of the brain through looking at connectivity and sharing of information. So we'll be applying that model to a brain network for face processing throughout this talk. And faces are an important and salient part of our visual world. One interesting part about faces is that we have different goals or outcomes related to processing faces. So for instance, you might want to know who a person is when you see them. You might want to make social judgments, like how attractive you think someone is when you see them. You might want to infer the emotions based on the facial expression. And you might want to take into account the dynamics. As over time, our faces move and our expressions move, so you might want to take the dynamics into account. So you might imagine that each of these functions that I've indicated for you are associated with a particular area of the brain. So the idea is that faces themselves are processed across many areas, so it's distributed, but each node processes a very particular function. And then all nodes share that information that each of them do. So for instance, if you take person identity here, you can imagine that information that begins in a region that processes who you are will then spread to all the other areas that might then use that information, for instance, to make social judgments or to derive emotions. And so we'll be applying this idea in our present work using functional magnetic resonance imaging to measure brain activity and connectivity of faces. And so I'll present to you 3 different studies. In the first study, we'll test our unified model of brain organization. We'll be removing shared information that's present between areas, and when we remove what's shared, we'll see what's unique in each area, and what's unique will be localized. In the second study, we're going to examine how connectivity can distribute representations. We'll be looking specifically at how person knowledge is associated with faces, and show that person knowledge tends to be localized in a particular area, but then that the connections end up distributing that person knowledge to other areas of the brain. And in the third study, we're going to be characterizing the representation of faces. And so we're really going to be focusing on this first point, the initial computations or operations that are done in a particular area. And we're going to be examining 2 such operations for faces. One, face identification, figuring out who you are. And the second, person identification, figuring out, sorry, I said who you are twice, figuring out who you are. And then in this first study, we'll be looking at how visual categories are represented. We'll be looking at faces, letters, fruits and vegetables, and vehicles. And we'll be measuring brain activity for various images associated with each of those categories. And we're going to be interested in this distinction between the localized and a distributed organization. Specifically, we're interested in our visual categories that we see. Are they localized in domain-specific brain areas, or are they distributed across a widespread array of regions? And we'll begin by looking at previous findings related to observing visual categories in the world. For the localized findings, people have tended to associate a particular area with a particular function. So for instance, for faces, they have an area that's called the fusiform face area. So even in the name, people have put the function that it is. There are other areas like the parahippocampal place area, which is associated with scenes and houses. It tends to activate for those categories, as well as for vehicles. And sometimes we'll also be talking about this area later as not a face area. So when you see faces, this area also gets suppressed. And then there's also the lateral occipital cortex, which is related to objects for which fruits and vegetables, which we'll be examining, are related to. And finally, we're looking at the visual word form area, which is associated with letter and word processing. Now, in contrast, when people have used multivariate pattern analysis, they tend to find more distributed findings. So for instance, if they're looking at where is information for faces processed when I show someone faces, they'll find that it's not just in this one area that I said, the FFA, but it's in all these different areas of the brain. So information for faces is present even in areas where we thought it was associated with words. So we'll be using these 4 different regions in our analyses that I described. And we'll also be looking at 3 additional regions that are primary sensory areas. They're regions that are control regions for us where we don't expect any category-specific effect. And in each area, we'll measure brain activity. And we'll associate that brain activity, which I've shown as a pattern here, with each of the categories. And we'll be talking about a measure I'll call classification or classification accuracy, which is the accuracy of predicting a particular category that someone is seeing based on the activity that's going on in the brain at a given moment. And so if you have a new activity pattern, you will use it to predict which category the person saw. In this case, you would say faces. And so we used regularized regression models, particularly the lasso, to run the analysis. But you can use other multivariate techniques as well, and they work sort of equally as well. And so now we were interested in how do we account for shared information in the brain. And our particular procedure looked at comparing a full model versus a left-out model, which here is where faces are left out. So what that means is you take all the activity patterns across all these areas of the brain that I've indicated, use all of them to say whether you saw a face or not, and then you compare that to all the areas -1, here this FFA area that's associated with faces, and you take the difference to indicate how much information for faces is present in the FFA while controlling for all the other areas. So if the FFA has a lot of information for faces over and above everyone else or all the other areas, then including it in the model will lead to higher classification accuracy. On the other hand, if the FFA has information for faces, if I look there, I'll find something about faces, but that information is being shared across or is common to many areas, when I compare the full model to the left-out model, I'll find a 0 sort of difference. And so I want to note that this analysis where I'm removing the shared information is similar for those in the field to including covariates in a regression analysis. For instance, including shared information from other regions can be thought of as including sort of motion parameters in your model. So I'm going to describe the predictions that we'll make. In this first slide, I'll just explain it, and then I'll give the predictions in the next slide. So what I'll be showing you is how the results for examining each region separately on the left. And then on the right, I'll be showing you examining regions together. So when we examine regions separately, what we're looking at is the activity pattern in a given area and seeing if we can predict what a person was looking at. Can we classify faces or not? When we look at regions together, we're going to be comparing the full model to the left-out model. And what we're left is the unique information in that area associated with faces. So now I'll show you 3 different scenarios that we'll be expecting here in terms of classifying faces. So first, I'll look at the scenario where the information for faces is in a particular area of the brain and is not shared. So in that case, both when you look at each region separately or you look at it together where you remove the shared information, you'll have the same result because the information was initially local to a particular area and it never really left it. Now, the second scenario is one that's very specific to our unified model, and it'll be a test specifically for our model. And so in that case, we have information that's initially localized, we're assuming to region A here, and then that information is shared via connections to all the other areas in the brain. So when I examine the regions separately, I get a very distributed representation because I'm getting the information in each area that's unique to that area, plus that's been shared from other areas of the brain. But when I look at the regions together and I remove that shared information with my procedure, I only get region A, because only region A has unique information associated with faces, and the other regions have only shared, and what's shared is removed. And then there is a third scenario that I think sometimes I forget and reviewers forget, which is that stuff can be initially distributed. So you can have a case where information for faces is actually present in every area, and it's unique in every area. And so that means if I look at them separately, each of them is going to have information for faces. And critically, if I look at them together, even though I'm removing this information that's shared between the three areas, I'm still going to get results because there's something unique and specific for each area that's relevant to processing faces. So here I'll show the results. So we have our regions that I'll show in different columns. And I've shown this scale here. So darker colors indicate higher accuracy in telling whether a face is there or not. So I'll start by looking at the analysis at looking at each region separately, meaning for each region we'll take the activity patterns and try to say whether it's a face or not. And what you can see here is that every region seems to have information for faces, and in fact every area is also statistically significant in having information for faces. And so that's even those regions that I said were these primary sensory areas where we don't think they do any processing for any of these categories, like A1, 2 is an auditory region. And so even this auditory region, based on this analysis, is saying, hey, there's something here in the brain, in the patterns of activity, that's specific for faces. So now if I contrast this with my together analysis, so I'm removing the shared information, I see a much more localized results. And so there's only 3 regions here that have information specific to faces. All the other areas now no longer do. And that's because if you notice in the last 3 columns, those were our control regions. So those control regions had information about faces in their activity patterns, but that information was being shared or in common with other areas. It wasn't specific to those regions. So now you might be wondering that face representation should be localized in the FFA. So I talked about in the localized model, there was this association between faces and this FFA region. So why am I getting these 2 other areas, which I call the PPA and the visual word form area? So to answer that question, I'm just going to focus on these 4 areas, the category-specific ones, and just plot back the results for the together analysis. And what I'll want you to note is that each of those boxes actually reflects an activity pattern that's predicting whether a face is seen or not. And in that activity pattern, there are other little boxes. So we often call them voxels, which are volume elements, 3D volume elements, from which we collect the data. And so each of those voxels in the brain can have a different direction. So you can notice there's a red color and a blue color. The red color is indicating those voxels in that pattern where higher activity is occurring when you see faces. The blue is indicating when lower activity is occurring when you see faces. And higher activity indicates that it's predictive of seeing a face, but lower activity is actually predictive of not seeing a face. And so what we can do now to try to understand what this result means is we can count the number of positive, so those voxels that have higher activity, and count the number of voxels that have negative activity in each of the 3 regions that had information for faces. And what we find if we move from the left to the right is in the FFA, you can see there's a lot of red, meaning that there's, of the activity pattern that was predictive for faces, there are voxels in that pattern that are actually related to, that are higher in their activity, predictive of seeing a face. So the FFA, like we've known in the literature, is selected for faces. And then for the PPA, most of the voxels in the pattern that's related to faces is actually not about a face. The activity in those patterns tends to be lower, and that's predictive of not seeing a face, as I said before. Now, there's a third case where we get a mixed result. So we get some of those elements of that pattern are predictive of seeing a face. There's higher activity, and some of those voxels are lower that are predictive of not seeing a face. So there's many things that can be said. I'll point out one thing that will be relevant to the later studies, which is that this visual word form area is actually very close to other areas that are relevant to faces. And I had mentioned that the faces are a distributed network, but the processing is very specific between areas. So later in the talk, I'll show how this FFA region and regions that are really adjacent and overlap with the visual word form area are both relevant to faces, but for different processes. And I think this supports our main unified model where information is initially localized to a particular area of the brain for faces, which here is the FFA and to some extent the visual word form area, and then that ends up being shared with the rest of the brain. And so if you look at each region separately, what you see is a distributed model. And briefly, I wanted to highlight the importance of considering the direction of the effect, since I think in the field people often don't do it. So here I'm just going to make a quick analogy to regression analyses for those who are in the field. And so you can imagine that when you do a classifier, it's very similar to doing a univariate f-test. But most of the time in the field when we do univariate brain activity measures, we actually do contrasts or t-tests, which is sort of analogous to me now looking at the direction of the effect. And we're more concerned with this contrast because it tells us something more meaningful. So for instance, on the right, you can see that there's two set of areas. One set of areas that is more active for faces than scenes, which in the field we say are face selective. And there's another set of areas that are more active when you see scenes than when you see faces. And so those are scene selective. So we found localized results after removing shared information. I wanted to note a couple of points. So one is that we also found the same results for the other visual categories, but I haven't shown it here. We also found very similar results for other regularization techniques like the ridge and the elastic net. And in study 2, we'll find the same pattern of results where if we remove the shared information, we'll get very localized results. So here we used a method to combine patterns of activity from different regions into one regularized model to remove shared information between regions. And this supports our unified model. So brain connectivity seems like it might encourage spreading of information, but category-specific computations are carried out in domain-specific regions. And I also provided some useful analogies that I hope some people might find helpful, which is that removing shared information is like including covariates in your analysis, your regression analysis, and considering the direction of these classifiers that we use in the field is like using contrasts in univariate analyses. So I'm going to now continue talking about faces, but a different aspect of face processing, which is person knowledge. That's in study 2 here. And so I'm going to use myself as an example. Greg had said he doesn't like cute stuff in slides. So I think this is my one thing that I'm allowed. So yeah, so this is it. So I should really milk it, I think. Okay, so if you imagine my face, you might imagine there's, so here it comes, there's facts that are associated with me, such as you might know that I'm a sensitive new age guy, a snag. You might know that my Facebook profile is really old, which my mom keeps asking me to change. And I work at SSS, the building behind this. And so even if you had never seen any of these pictures and you just know those facts, you might say, oh yeah, that's Zarrar, and it might prime an image of me in your head. So you can see that faces are really associated with knowledge about people. Okay, so we're going to ask the same question if person knowledge is localized or distributed. And specifically here, we're interested in if person knowledge is localized to domain-specific areas or it's distributed in various domain general areas. And remember again that faces are part of a distributed network. And what I'll talk about now is how, even though it's distributed, it can still be consistent with this localized sort of single area model. And that's because you can have each region of a distributed network be related to a specific process. So here I've shown 4 regions, and they work in a feed-forward matter, where on the left hand, at the beginning, you have face parts that are processed in the occipital face area, and then you have the whole face that's being processed in the fusiform face area, or sometimes we also say the FFA processes perceptual information for faces. And then the other 2 areas, which I'll tend to group together when I talk about them due to their shared function, the anterior fusiform and the VATL, both are related to person identity and person knowledge. And these 4 regions tend to also have potentially differing functions depending on the hemisphere you're looking at. So here, and as I go forward, I'm going to be showing the brain where on the left-hand side is the right hemisphere, and on the right-hand side is the left hemisphere. It's just the convention we use in the field. So in the right hemisphere, faces are typically, most of the studies find that when you show people faces, you get activity mostly in the right hemisphere. But there's some work that finds that there's a left lateralization for face naming and face memory. And this will be relevant for the current study because we're looking at person knowledge, which is related to face memory. And in contrast, there have been some work that have found a more distributed representation for person knowledge. So they posit that information content is not defined by the area. And so when they look at person knowledge, they find that the person knowledge is distributed, not just in the VATL, but also in this FFA area. And so the FFA area that I spoke about can then have multiple functions. So it's both about knowledge about a person and about perceptual processing of the whole face. Here we seek support for our unified perspective. We made an assumption that information is initially localized, so the FFA is about perceptual processing, and the other 2 areas about person identity and person knowledge. And then that local information is then shared. For instance, the FFA sends perceptual information forward, and these other two regions send information back about person knowledge. And that spreading of information occurs not just between these two information, but the VATL is spreading information throughout the brain. So what we end up getting is a very distributed representation for person knowledge that we end up observing. So that's our unified model. And we'll seek to get support for that with 4 analytical techniques. And what we'll be asking participants to do is associating 3 different facts with each face. And with half of the faces, they'll associate biographical facts. So like, what is a person's name? They'll learn that association with their face. And with the other half of the faces, they'll learn physical facts, or like perceptual facts, like what is someone's eye color. And then in the fMRI scanner, we look at brain activity associated with participants viewing each of those faces while recalling a particular fact with them. When we look at the results, we'll pay attention to a particular contrast here, this contrast of faces associated with biographical facts when compared with faces associated with perceptual facts. And I'll also talk about that as the biographical condition versus the physical condition. And that's really going to get us activity associated with person knowledge by taking the difference in activity between those two conditions. So I'll start by looking at these univariate brain activation results, where I'll look at a particular slice of the brain. And here I've plotted the brain activity along that slice. And what we see is that person knowledge is localized in the VATL and anterior fusiform, particularly in this left hemisphere that I mentioned was related to face naming. And we don't see any effects in the FFA or the OFA. There are other regions that are relevant, like in the early in visual areas. We won't be talking about them now, but I just did want to focus on that the right hemisphere here has no association with person knowledge for any of those regions. So really these effects in the 4 areas we were interested in are specific to the left hemisphere and specific to this VATL and anterior fusiform area, which supports a localized view as well as our unified model, where we're predicting that person knowledge is specifically associated with the VATL and anterior fusiform. In the second analysis, we looked at multivariate brain activation patterns like we did in the previous study with visual categories. But here we're going to be classifying whether a particular activation pattern is related to the biographical, the faces associated with biographical knowledge, or the faces associated with physical knowledge. And we have the same scale there, where darker colors indicate that you're better at the activity pattern is better at discriminating those two conditions. And I'll be using the same 4 areas that we've been looking at in this particular study across the 2 hemispheres. So first we looked at the results when we looked at the activity patterns for each region separately. And what you can see here is that there's information from person knowledge in every single area, and those results are significant in every single area. But when we look at the results together and remove the shared information, we can see a much more localized representation. So now all these areas in the right hemisphere that had information for person knowledge based on their patterns and activity no longer do, because that information wasn't unique to those areas. It was shared between areas. And of these 3 regions we have left, well, 2 of them were significant, the left FFA and the left anterior fusiform. And so one thing could be that we haven't looked at the direction of effect in this particular study. So maybe that the information in the FFA here is actually not about that biographical knowledge, the person's name being associated with the face, but more about that physical knowledge, like someone's eye color. And so we might need to do those tests later. The second idea might be that information in this anterior fusiform and the VATL process person knowledge and then feed it back to the FFA. And then the FFA does additional processing after it knows who it is. It does more processing, which is unique to that activity pattern. And so we'll be exploring that idea, the idea of feedback, in this, the idea of feedback and the idea of sharing of information in the next 2 analyses. So in the third analysis, we look at whole brain connectivity. We measure connections for each region of the brain with the rest of the brain. And then we count the number of connections where the connectivity between 2 areas is stronger in the biographical condition than in the physical condition, meaning that connection is communicating person knowledge. And what we find is that the VATL and anterior fusiform, which I'll focus on here, have the most connections with the rest of the brain that discriminate between the biographical and physical condition. In other words, this VATL and anterior fusiform area in the left hemisphere, by the way, is a hub for person knowledge, and it's distributing person knowledge throughout the brain. So this really supports our unified model, where information is localized in a particular area and then spreads throughout the brain. We then explored directed connectivity analysis, where we looked at the more specific, the directions of the connectivity. So before, we can't tell if the VATL is feeding information to the other areas or the other areas are feeding it back. And so we explored that idea here. So I just wanted to give you an intuition for that analysis here by giving an example of clouds causing rain. So imagine, so to answer that question, let's imagine different scenarios. So imagine that how often do clouds occur? That might be 30% of the time. How much do clouds and rain co-occur together? And you might say, oh, maybe that's like 20% of the time where I live. And how often, and this is sort of the critical aspect, how often does rain occur on its own, right? And hopefully you would say never. And then if you say never, then you could see that rain is dependent on clouds. So the occurrence of rain is dependent on the occurrence of clouds. And that dependency in these sorts of analyses is used to make the inference that clouds then cause rain. So we're going to apply that same idea to our different brain regions. And so here I've shown 3 of the 4 brain areas. The one area that I talked about, the anterior fusiform, didn't have any additional information, at least to show visually, so I dropped it out. So what we see is we have these 3 areas, and the numbers indicate the direction of the relationship. The star indicates that it's significant, and the grayed out voxels indicate that it's not significant. And what we can see first is that there is an increase in feedback signal from the VATL to the FFA for the biographical versus a physical response, which means that there's information about that person that goes from this VATL area and feeds back to the FFA. And we also find an increase in a feed-forward signal from the OFA to the FFA for the opposite contrast, for the physical greater than a biographical response. And what that is meaning is that perceptual information about the face, the visuals of the face, seem to go from the OFA up to the FFA. And so this, again, supports a unified model where information is initially localized, both for person knowledge and perceptual knowledge, and then shared with other areas. So we can draw this study together and ask, where is the information for person knowledge? And it seems to depend on the particular method that we were using. So the brain activity methods seem to be showing something that was more localized, whereas the classification methods seem to be showing stuff that was slightly more distributed. But I think if you take all of them together, all of these findings together, you probably see support for our unified model where stuff might be initially localized, then it spreads throughout the brain, and then it looks like it's distributed. So in study 3, we're going to continue to look at face processing, but now we're going to try to understand what the particular mechanisms are. And we'll be building on the ideas we were looking at in the first 2 studies. So in the first study, we looked at face identification. So we were interested in how activity patterns said whether or not there was a face there. In the study 2, we looked at person knowledge, which is a part of how we identify who a person is. And so we'll be looking at both of these ideas here in this study, how face identification works and how person identification works in the brain. So for face identification, and for both of these ideas, we'll be borrowing work from computer science and the psychology literature. And so for this first point here, we're going to be trying to understand how we can tell that there's a face there. And the way we're going to measure that is we're going to compare each image that we see to the average of all the possible faces, which is shown on the left. And so if an image is closer to the average face, the more it will look like a face. And the further it is from that average face, the more it won't look like a face. In fact, maybe it won't even be a face. And we'll use the same idea for person identification. And here we're thinking that for each person, we have some template. For people we know, we have some templates of what they look like in our head. And that template is reflected by the average of that particular person's face. So the average of all the times we've seen them. So here I've shown Angelina Jolie. And on the left-hand side, you can see one possible average of her face. And what I can do is compare each and every photo to this average. And so photos that look more like Angelina Jolie photos that are closer to Angelina Jolie then are inferred to look more like her. And photos that are further away look less like her, and in fact might even be a different person, like shown here under the blue circle. And we did an fMRI study where we showed participants stimuli of 8 different celebrities. for each of the celebrities, we showed them a variety of photos. And you can see really the diversity of the photos that the participants saw as we were measuring brain activity for each of them. And so you can see they differ quite a bit in pose, likeness, expression, and age. And for those who don't know, this is also Angelina Jolie. And so if we're trying to then make a comparison between the different images here, they differ so much, it'll be difficult. In fact, if you just try to like point where the eyes are in a different spot in every image. And so that's going to be a problem if we're trying to model how the brain works. And so we're going to need a way in which we can put these images in some space where we can compare them based on who they are rather than what the image actually is. And we did that, we addressed that issue using a neural network. And the way the neural network worked is it was trained on hundreds of thousands of faces to tell if a pair of faces were the same or were different. And then what that allowed us to do is take any new image and transform it into 128 values. Each of those values indicates some aspect of that person's face. And then we can put those values in a 128 dimensional space, which we call a face space. Here we'll just be visualizing that face space in 2D. So what's really nice about the way that works, that process works, is the neural network ends up allowing us to map faces that are similar to each other, closer together, and faces that are dissimilar to each other, maybe even of different people, will be plotted further away. And we can use that representation of faces that we get from the neural network in this face space to get our face-like and person-like measures. So each of the circles here shows a different photo. The blue indicates the center average, which is the average of all faces, which is important for face identification. And in pink is the person average, the average of that particular person's face. So if we take one of the photos, we can get the distance to the center average, and we can get the distance to the person average. So photos that are closer to the center average look more like a face, and photos that are closer to that person average look more like a person. So that's how that analysis works. Now, when we're going to be looking at brain activity, there's two possible predictions that we can make about the relationship here. One is this prediction error model, where higher activity occurs when a photo is further from an average face. And so what that means is that in our heads, we have some expectation of what we're going to see. So our expectation is, oh, I'm going to see the average face of Angelina Jolie. And then if the face that we actually end up seeing is a mismatch, it's a different face, then you'll get higher activity. So that's that idea. And then there's the template matching model, which is that higher activity occurs when you're closer to the average face. And so this is more like you have some template or some filter in your head, and everything that comes in is trying to be matched to it. And the only time you care is when it's a good match. So like when the thing actually looks like the average face, then you'll really activate. So now we'll look at our two different results here. First, I'll look at the comparison of the photos to the person average. We find that higher activity occurs for faces that look more like the center average, and this happens in this right fusiform area. So, and this is the area that we got in the other two studies as well. And the photos, comparing the photos for the person average, occurred in the left lateralized regions and were related to higher activity for faces that looked less like the person average. And so those areas, remember, we also got when we looked at our person knowledge effect. And so these results, I think, really support a localized representation where the center average is related to one area and the person average is related to another set of areas. And if we think about these results in relation to the predictions that we made, the comparison of photos to the center average reflects a template matching model. So this is almost like when we walk into the world, we're always trying to see faces, faces in clouds, faces in food. And so that fits with the template matching model, right? So you're in somewhere in your brain, it's constantly trying to see whether there's a face or not. And when it thinks it sees the face, suddenly you're clued into it, right? So that really supports that. And it also supports this idea that faces tend to activate the right hemisphere, which is a lot of the literature finds. Now, for the person average effect, we see something more consistent with a prediction error model. And this might be more like the idea that when I'm expecting to see someone, I have some expectation of them, and that expectation is related to this average of every time I've seen them. It's like this template. And if they don't match with it, so maybe their hair is all different, you might be shocked and be like, oh my God. But if they look exactly like you expect, nothing changes. You would go about your day as normal, so there wouldn't be much, say, brain activity as an analogy. And just one point to make here is that faces, that we actually also got a relationship between this brain activity measure and when we asked participants how similar were these photos to the person. So if participants were asked for each photo, how much does this look like that person, then that actually related to the distance between the person average and that photo. So photos that were closer to the person average were rated by people to be more like that person. So to summarize our 2 localized face computations, we found one was face likeness, which reflected this template matching model, seeing faces everywhere, and was localized to the right FFA. And in the second effect, we looked at the person likeness measure, which reflected a prediction error model, where we had some expectation of what face we wanted to see, which was localized in more in this left lateralized FFA and anterior fusiform. And the one point there's a lot of points one can make on the study, and the one thing I wanted to focus on was the types of stimuli we used and what it might say about the field in general. So usually in the literature, you look at between people. So when they measure brain activity, they look at how that activity relates between different people. Whereas what we did here, which is fairly unique, is to look within a person at the different photos of that person and how those photos might relate to each other. And how is it that even though we have all this variability in these photos, how can we recognize that person in spite of that variability? And so maybe the literature that tends to look at between people identity finds effects in the right hemisphere. And so I'm just speculating that maybe in this left hemisphere area that we tend to get, it's more about within that person, like what I know about that person, how I can overcome all these differences that I view of that person over time and view them as one person. And so maybe the left hemisphere is about the within person effect. So I'll just quickly bring it all together here. So we showed in these 3 studies this model where information is initially localized, then shared throughout the brain, and the effect of that sharing is what we observe as being very distributed. Sorry, I'm supposed to skip those slides. So now I just want to make 2 main points. So the first point I wanted to make, and both of those points will be about shared information, since this is often the thing people are interested in. Well, you're removing all the shared information, focusing on what's unique, so tell us a little bit more about shared. So the first thing I wanted to say was that people don't really remove shared information or consider the direction of effect in our field, and suggest that it might be an important thing to do. It might lead to spurious results, but if we do it, we can also maybe get a richer idea of what the effects are by seeing, well, where is the information? Is it in one area or is it shared between areas? And that additional insight can tell us more about brain organization. And then the second point I want to focus on, what is the nature of this shared information? So there's 4 points I was going to go through, 4 different ideas that might explain what's going on. So one is that the shared information reflects inputs that come from another area that will be processed locally. So for instance, I talked about this occipital face area that processes parts of the face. So maybe that's processing part of the image that you're seeing, the face parts, and then feeding it forward to this FFA area that does a perceptual processing. So that takes as inputs the processing of the OFA and then does something additional on it. So you would have information in this FFA area for both the FFA's processing and what's being shared from the OFA. The second idea is that maybe just the way the brain works is it just sort of has an obligatory spreading of information. So one idea we had thought about in the lab for this was that it's like when I speak. So the information is initially coming from my mouth, and then the sound waves are gradually spreading throughout the room. Without anything happening, it's just spreading. In fact, it's even vibrating, the sound waves vibrate on the wall. And so in that same way, information from one area just sort of spreads throughout the brain, and the expectation is that the other areas that are concerned with that information will process it, the regions that aren't will ignore it. The other idea that's related to this prediction error model that I had talked about before is that the shared information may reflect something that you're preparing for in advance. So maybe if I hear someone's voice, I might have other visual areas in the brain, and I share that sort of voice information with those visual areas in advance, so that in case I see their face, I'm like ready for it. So this is sort of preparatory. And the one that I find very interesting is this idea that actually, even though the same exact information can be shared between 2 different areas, they have to then be transformed into another way of looking at the same exact data. And that transformation offers different perspectives in different brain areas at looking at the same data. And it's the same idea, actually, of of those different brain areas possibly offering different perspectives that I was hoping to take in the work that I showed you now. So to connect that idea here, where I use various different methods to get at different perspectives on the data, to overcome any weaknesses of one method, but also to offer us a more complete understanding of what's going on. And that might, I hope, address these issues where specific methods end up giving rise to specific brain organizations. So that's the end of the talk. I have just some acknowledgments that we tend to do. I wrote a bunch of Post-its because I thought my talk went a little over last time, so I wanted to make sure I didn't go over. So, yeah. So I was going to start with Marcia. So Marcia was on my committee for a lot of these projects, and she sort of helped think through a lot of these projects. We also had, for some of you here will remember, we had a writing group, and I found like all my interactions with Marcia were like super. The thing I remember about it is that whenever I would meet with her, with like writing group that we had, or privately, I would leave the meeting being super positive, and also being like, I understand my like work. But it was scary because then it was like one of those memory loss. I would lose that feeling of that understanding, and I'd be like, no, what's happening? What's going on? And so and I would keep thinking like, oh, and this is the other point I want to make. Oh, what did Marcia do to think about this in that way that led me to that understanding? And I feel like the one thing was that interacting with Marcia, she was like the most, in psychology we have like a word like growth-oriented, which means like effortful, you focus on effort and being future-oriented, and she's like the most growth-oriented person I've ever, like I'll complain and she'll just like, I don't think she ignores it, she's just thinking about the effort I should put in and the future that needs to be done, and she just ignores it, but just talking about the things I can actually do. And so I think I really took that away from Marcia, is that really that you can do stuff yourself, you can think about these things yourself, and as she often says, writing is thinking. So I feel like as I was going through the introduction and discussion of this dissertation, I was like, yeah, I was thinking about these ideas, but as I wrote them, they really connected together. So thank you to Marcia. And then Marvin is next. Everyone uses this picture now. That he has this portrait that was made. So Marvin's also been with me through all these different studies, and I have a different Post-it for Marvin. Yes, so, right. So the one thing I want to say was that I often appreciated Marvin's very incisive comments, and very like, his comments like, we're always considering the whole field, like, and where you should go for your career, all in like one sort of comment. So I definitely appreciated those, and I liked that how they helped me think. The one thing I also liked about Marvin in relation to those incisive comments, for some of you here, you might know we used to have like state of the lab addresses that Marvin gave. And they were very intense affairs. There was a lot of controversy about it, but I really appreciated it. Marvin talked about what the lab was doing, but he really emphasized that if you wanted to succeed in this career, there were things you needed to do, papers you needed to publish, work that you needed to do if you wanted to get sort of the job that maybe Marvin had, or the life that Marvin had. So again, I felt like there's a theme here. There was also this very growth-oriented thing. It was like what you did really mattered, and you really had to work. And it was nice because for me it was like, oh, it really made me think, because normally you just travel through the process. And so both Marvin's private meetings and those lab meetings were helpful in being like, oh, I should really think about this. Do I really want to keep going? Because it's a lot of work. And so I think it was helpful getting that perspective into account. And so I have Greg now. I was wondering, like, this might be the only time I get to talk about Greg in this way. So I need to savor it. So, yeah, so I wanted to start by saying, at my maybe first year working with Greg, I was at a conference with Kia. And I met Kia, and I told Kia, you know, some people think Greg's really scary. Like Kevin Pelfrey said that Greg's really scary. And I'm telling this to Kia, and Kia looks at me, she's like, no! And I don't remember the words he said, but I think it was something like, he's a sweetheart. And so I was like, sweetheart? And I'm like, well, I mean, Kia would know, right? She's experienced. And I actually feel like through this process, I actually do feel like that now. Yeah, I've learned something about this where Greg will seem like he's like really upset or something, or like he doesn't want to deal with those students, or like with me, he doesn't want to deal with me. But actually, it's crazy. He spends all this time with teaching. He spends a lot of time with students. He spends a lot of time with me. So I realized, oh yeah, Keo was right. He is really. I can't say the word again. I should have found out what Key actually said. Maybe it's something totally like different. And then, but in that regard, I also appreciated that Greg had very blunt comments to give. So at lab meeting once, he told me when I gave a practice talk, not this one, he said, "Oh, I almost forgot how terrible you used to be." And I think other people were horrified. And I, again, like I've been mentioning, Marcia, Marvin, growth-oriented. I was like, oh, so I was at the bottom before, and now I'm awesome. Look at that difference. And I actually practiced a lot on the talk. I felt really happy, actually, about it. So I appreciated those comments because it made me feel, okay, I can work and I can get better. And then when Greg says, oh, it's good, then it means it's like amazing. And the last point I was going to make is that I felt, like with all of grad school, that with Greg, I really learned how to think, like in our lab meetings, in our private discussions. Like we'd be done with the research, and suddenly he's talking about evolution. And I'm like, I don't know how evolution works. And he's just looking at me, and we're talking, and it's interesting. I'm like, oh, I am interested in it. And I really feel like from that, I have this real appreciation of now like looking at articles and anything, in not just in research, but anything, I'll look at it with this other light and think about it deeply. And so also I wanted to thank the rest of my committee for the comments they've given me and the random times we meet in the hallways and maybe encourage me or tell me how stuff is going to be bad. But yeah, so thank you very much. It was helpful. And I wasn't going to talk about my old advisors that much, Mike and Cameron, but I'm told they're live streaming. So in case they are, I thought I'd speak about them a little bit more. So that's Mike on top, and that's Cameron. They're in New York, although Cameron's now going to get a job at University of Texas, Austin. And that's sort of the rest of the team that I worked with there, Javier and Adriana. I had another card here. That's where it was. Okay, so I mean, there's a lot I could say, but it was a helpful time that I worked with them. They're a much bigger team. I feel like it's funny when you work with someone, you're often like, oh, I don't want to be like them. You're like with your parents too, you're like this. And then later, you're just like with both Mike and Cameron, I feel like their thinking style and the way they work is now how I sometimes do things, like how I write. Mike has a certain style of writing, and now I'm like wanting to do his style of writing. And then I'm like cursing myself. I'm like, no, what have I become? But I think it's been, so that's what's really nice for me is that they've really helped me, and their style of thinking has really been integrated into the way that I work. So a lot of the connectivity analyses that I'm showing, a lot of the methods, although they haven't directly worked on them, I've worked on those things in the past with them, and a lot of that thinking is sort of implicit throughout those studies. And I also had the fortunate time of having a lot of collaborations with people from the statistics department. There's Phil Reese, who I worked with before, but I just wanted to mention Jay Emerson and Joe Chang, since they're here at Yale. It was nice, I was able to go there, chat with them. With Jay Emerson, I basically learned how to code in stats, and it was very, very helpful. And they're very intense, but it's good, because it's very growth-oriented. So I learned a lot about thinking about things in a statistical way and about coding things. And then as Greg mentioned, I started here with Jeremy Gray, and it was like really great. It was almost a separate life that I lived. And what I liked about Jeremy is his very sort of interdisciplinary way of thinking and also his sort of life. So here we're using a door that was like a regular door here to make a table. So this idea of like, oh, I wouldn't have thought to use a door as a table. And so that's what we're doing. So apparently the emergents, the ones that are for fire safety are really thick, and so they are really good wood. So they actually would make good tables. And of course, thanks to all my friends and family. And I had one last point that I was going to make. So there's two sub-points. That's going to be funny. So last night, they're both related to my mom, and a story related to this and being in a PhD program. So the first thing is my mom last night was asking me, if I get bored, is it okay if I like leave and take a nap and come back? And then my brother is just like feeling fine. He's like, announce it publicly. That, you know, but with that in mind, that's my mom. When I was a kid, so this is not a joke. It's so weird. I remember this again very recently. And she remembered it too. So when I was a kid, she would tell me if I was like playing with toys and wouldn't come for dinner or wouldn't go with her to somewhere else, she would say, "It's not like you're doing your PhD." And she stopped saying it at some point. I don't know why she stopped, but I don't even know why she said it. But she did, and so yeah, I guess now I've been doing my PhD and now I'm done. Yes, yeah. Thanks.
So we have time for a few questions.
Yes.
I have two questions. But one is at the very beginning you said each region can accept any information.
Yeah.
But you weren't really showing that each region accepts any information. Don't you think there's some specificity in what information each region can accept? I understand the main point.
Well, I guess there's two levels. So at the end, I talked about something I don't exactly know, which is it's possible that it accepts any information in the sense that sound propagates through the room, and you may either attend to it or not attend to it, but it's still like going through the room and spreading. So in that sense, it can accept any information in the sense that no matter what it is, it can get in there, into that region. But then the second thing I guess you're talking about is, yeah, something can select for it. So you may not.
It depends on what we mean by accept.
Yeah. Well, by accept I mean that it processes it, yeah. So then that area is going to say, oh, okay, I'm going to do something more with this information.
Yeah, so you mean it, and like the wall does accept the sound?
Yes. Yeah, so the wall does.
It doesn't do a lot.
Yes, that's right. Yes.
Okay, the other, your person average, whether a region is getting more active or less active, whether it's a prediction error or a template.
Yeah.
Does that depend on people in your particular experiment? People having the class of possible faces kind of potentially active. In other words, if you get the prediction error, would you get a prediction error for that kind of information if they didn't have a limited set of faces it could be?
Yeah, and so we also told them, so to give a background on the task, so they were shown 8 different celebrities, but they were also shown some distractor faces. There were about 6 of them, I think. And for those 8 celebrities, they had to press a button for which celebrity they saw. Since there's 8, we split it into 2 chunks of 4. So at any given moment, they were pressing a button for 1 of 4. So like you're mentioning, they were probably primed for it.
Right.
So I think that's probably true, that if they hadn't been primed for it, they have no expectation. And so the process may work to do it differently.
Right. So, but you still, when you're not expecting any particular person, but you get something that activates person information that might feed back and help you recognize that person at that face.
Yeah.
You would get that, but you might not get it showing up as prediction error in your signal. It might show up as greater activity relative to some reasonable control.
Right, exactly. So with our data, we may not be able to look at those specifics. So the specifics you're talking about is like, I see a face, I don't expect to see it, and say it's because Marvin's looking at me. I suddenly see Marvin in the street, and I'm not expecting him. And then what might happen is it might ping to this person area that says, oh, it's Marvin, that then pings back to say focus on it or do something. So yeah, I think that's how I would think it would work. So it's working in these different parts and they connect together. But yeah, I think in our particular data, we may not be able to see it because the fMRI has a sluggish response.
So the prediction, I guess my more general point is the prediction error might be a more specific thing that only operates when an agenda is actually...
No, so the idea would be that we're looking at a particular part of this process. So even if I didn't expect to see Marvin and suddenly I see him, and then I activate some region, I'm still positing, although I don't have the data, but I'm positing that then I have a prediction, and that's the average phase, and that gets sent back. Yeah.
Great talk.
Thanks.
One more question?
Just thinking out loud, so at one stage you suggested that the OFA would pass information to the FFA. Do you think, or is this also reflected in other methods, like with integrating recordings? Do you see that the OFA is active temporarily earlier than the FFA?
Yeah, actually the results are different both with intracranial recordings, and there's an fMRI paper that looks at this. And they find that actually the FFA activates earlier than the OFA, with maybe some idea that some information is going forward, and then the FFA is like, hey, it's a face, and then it goes backwards. So maybe my example isn't totally accurate, but it goes backwards. But I guess you're asking with other literature. Yeah, there's been some, but I feel like in the intracranial, there isn't that much in terms of this, looking at it in this way. Information is initially localized and then it spreads.
I have a question. To what extent do you think this would generalize beyond face processing? So I imagine you chose that for a specific reason.
Yeah. So we found it for the other visual categories. So each of them you find the same type of process. I did it with faces, but that's with person knowledge, so it's not exactly with faces, and it generalized. So I feel like it should generalize because it's a basic principle that I think most people, so I didn't say this in the talk, but I feel like most people I talk to have the same implicit like unified model that I spoke of, where they think that things are specifically done in particular areas and then it's sort of shared between areas. So I think since most of us seem to have that also as an implicit assumption, I would presume these results will work with any stimuli, any function. The second point I wanted to make though was that you can also use different classifiers, right? So imagine with a face you had I showed those many different faces. So I could look at pose, I haven't done this yet, but I want to. You can look at pose, expressions, and with each of them, you can use a different classifier. So each classifier is picking up in different types of information, and by picking on different types of information, you might get different localization. So.
Okay, great. I guess I just wanted to have this opportunity to give another state of the department. And this is mainly for the graduate students here. I'm sorry it's unsolicited, but this is exceptional. Zarrar has been an exceptional student of our program, and we should strive to be like him. He has been incredibly productive. He is technically sophisticated. He does conceptually novel things, and I just wanted to add also that he's been an exceptional colleague. I think many of you really appreciated his expertise and just how much fun it has been to discuss ideas with him. And so this really sets a high standard. I'm still not sure if he's going to pass or not, but I had to share that. I had to echo what his sweetheart had started. So let's thank him. So please wait outside or in the lab at the reception, and we'll go into private session.